Good morning, everyone. So let's go ahead and get started. I'm Tarun. I'm the chair of the Early Career Committee for SMRA. And welcome to our plenary session. Um, so the ECC is the primary trainee committee. As for the SMRA, we do a lot related to career development, as the name would imply, for our trainee members in the SMRA. The committee is made up of trainee and some faculty members. Our trainees we primarily organize the SMRA webinars and this conference plenary session. We also publicize MRA related research on our social media platforms. And if you're not subscribed or following those, you should. We also liaise with the other SMRA committees, including the Education Committee, Communications Committee, and the SMRA Board. And we're always on the lookout for more friends and to learn more from our esteemed mentors. And for those of you in the faculty, we'd always love to hear from your career experience and to help you connect more to our next generation of MRA researchers. So for this year, our initiatives mostly focus on our monthly meetings, and we also have the opportunities to connect with the other committee meeting, or we welcome new members all the time. You can just email me if you're interested, or email any of our admin staff. We also organize the webinars and this annual career plenary session. And our current initiatives mostly involve the Junior Fellowship Award and the networking coffee break, which will be happening right after this. So our current members, I'm the chair. Our past chair is Calder Shigrin. He couldn't be here this year. Our current faculty advisors are Suzanne Schnell and Jihai Zhao. And our committee liaisons, Judith Shurd from Semmelweis, Education Committee, Mahmoud Mosabasa, and our social media lead is G.H. Chang. So yeah, if you're interested in joining, please contact us by email or just get in touch with one of us, and we'll be happy to add you to our email list. So now I'm going to hand it over to the SMRA president to introduce our junior fellowship winners.
Thanks, Tarun. Good morning, everybody. It's my distinct honor to talk a little bit about the SMRA Junior Fellowship itself, as well as then introduce the 2025 awardees. So what is it? This was a major initiative from the Early Career Committee that was initially kicked off and discussed extensively in 2023. And the first year the award was held and applications were accepted and the awards were handed out was last year in Chile in 2024. The opportunities available to junior members of the SMRA. What are the goals? The goal is to recognize early career postdoc and clinical fellow MRA researchers for contributions to the field. And also a second, maybe a little more self-serving goal is to recruit such individuals to the society and to encourage and reward societal involvement earlier in their careers. The eligibility requirements, we're looking for individuals earlier in their career, so 35 years old or younger. They need to hold a postgraduate medical degree, a PhD or equivalent doctoral degree. And they need to be enrolled in a training program, a residency program or a clinical fellowship program, or working as a postdoctoral researcher, or because of the differences in training across all of our sites within 3 years of their first academic appointment at the time of the application. So we're looking for early career individuals still in training or just completed training from a clinical background or a science background. What are the benefits of being awarded one of the junior fellowships? Well, there's a travel stipend to attend the SMRA annual meeting. They get to present at this session, the Meet the Junior Fellow session in the Early Career Plenary. There's a trainee board membership as well as an invitation that they receive to the on-site board meeting. It's typically held on the third day of the meeting. Today actually is the board meeting, so they'll have an invitation to that. The biographical interview that they participate in and provide information to will be uploaded to the SMRA website and posted on social media. And they are paired with a senior SMRA mentor who has a similar research area. You can find more details on the Society website if you follow this QR code. Well, I want to thank the awards committee. The awards committee was stood up to help us review applications and suggest who the award winners would be. So I just want to call out those members here. Thank you again to Oliver, Shihai, Susanna, Alexandra, Calder, and Anastasia for their work. This took a lot of time to review the applications and discuss and determine who the awardees would be. And so without further ado, the 2025 SMRA Junior Fellow Award winners are Thekla Oktering from the University of Madison, Wisconsin, and Suvai Gundasekaran from Cedars-Sinai Medical Center. So congratulations to both of them. And Thekla was going to attend in person this year, but was unable to at the last moment, so she sent a recorded lecture. Suvai has perhaps the best reason to not be in attendance at a meeting in person ever. She's getting married this weekend, so she's going to visit us next year in China. And then Lexi Fan, who was one of the awardees in 2024, but was unable to attend that meeting, was intending to join us here in person. She's also going to deliver her talk today, but it's also going to be recorded. So we'll kick those off.
Dear colleagues, mentors, and friends, dear members of the Society for Magnetic Resonance and Geography, thank you so much for this incredible honor. Being selected as a junior fellow means a lot to me, and I am truly grateful for this recognition and support. MR in geography is such a fascinating and important field. I feel lucky to be part of a community that's pushing the boundaries of what MR and geography can do for patient care. And this motivates me even more to asking questions, to exploring new ideas, and to collaborate with members of the society who are just as passionate about MR and geography, and in my case, especially 4D flow MRI. Thank you so much for giving me the opportunity to introduce myself and my research to the society. I grew up in Leipzig, which is south of Berlin. My parents are both vets, and I experienced how much fun working and exploring together can be. Apparently, the X-ray table was the most convenient spot to put me on while my mom was seeing animal patients in her clinic. My dad stayed at the university and became the head of the small animal hospital at the University of Leipzig. He made my school project on X-rays possible. When I was 15 years old, we examined my pencil case with conventional X-rays and CT. I wrote an essay about the physics and technology behind it, the benefits and drawbacks of X-ray and CT. That is when research got to me, and I knew that I always wanted to do research, learning about new things and applying them to new problems. In this case, the contents of my pencil case. I couldn't get enough and did another small research project in the following year. Again, I was focusing on medical imaging. I learned about conventional and CT virtual endoscopy using my recorder, a bell pepper, and a clay flute as study cases. My dad and my mom often discussed their research at home, and they kind of infected me with their fascination. I asked if I could contribute somehow and do a real research project. I worked together with a small animal anatomist and studied the anatomy of brachycephalic dog breeds in CT. These dogs have trouble breathing because although breathing shortened their noses, the structures within their noses did not get the message and still try to grow as if they would be living in a large house instead of a one-room apartment. That gets very crowded and the pugs often have problems breathing. On the left, you can see the coronal anatomy in a German Shepherd dog for comparison. On the right, you can see the tiny nasal cavities of a pug. We found that in many brachycephalic dogs, conchae grow into the nasal meatus, obstructing the airways. You can see it here and there. This should be free as it is here. At the University of Leipzig, they have developed a unique endoscopic surgery technique to help these animals breathe again. They treat animal patients from all over the world with long waiting lists. I would be more than happy to discuss the implications of wrong breeding standards on animal welfare another time. I studied medicine in Lübeck because the med school had the best students' ratings in Germany. In the radiology course, which I of course found fascinating, I also met my husband, Nicholas. We tried for the rest of our studies to convince each other not to do radiology because we thought that would be weird working together in the same department. Well, we failed, and we've been working together ever since. The same time I fell in love with Nicholas, I also fell in love with 4D flow MRI, and I started working on my doctoral thesis. I was captivated by the method and its potential. Alex Frederikovich was my mentor and supervisor of my thesis. Professor Sievers, who established the Sievers classification for bicuspid aortic valves, was the head of the cardiac surgery department, and we met frequently to discuss post-surgical aortic hemodynamics and potential surgical improvements. The problem with routine valve-sparing aneurysm repair with a straight prosthesis is that the valve leaflets hit the prosthesis wall during systole. Professor Sievers had developed a sinus prosthesis that promised normal sinus vortex formation, which is important for proper valve function and optimally would enhance valve longevity. We were able to visualize sinus vortices in normal subjects, and we found similar vortices in the sinus prosthesis. This stands in stark contrast to normal tubular prosthesis where no normal vortices developed. For this work, I received the Thesis Award of the German Röntgen Society. Nicholas and I stayed in Lübeck for our residency, and I continued my research and was accepted into the prestigious clinical scientist program at the university that included dedicated research time, my absolute dream. With the support of my department chair, Jörg Barkhausen, and Alex Frederikovich, I gradually took the lead of the growing 4D flow MRI research group in our department. We continued studying Professor Sievers' patients. Although the flow in the aortic root looked great, we found disturbed flow distal to the prosthesis. This was presumed to be due to kinking and post-prosthetic dilatation. No wonder. We are implanting straight tubes into a curved vessel. Professor Sievers had seen that, and he had developed a 90-degree pre-curved prosthesis that should reduce the kinking. This was unfortunately only the case in 1 of 10 patients with this prosthesis. But this one patient had no kinking and normal flow. And this underlines the importance of normal anatomy to allow for normal flow. With other doctoral students, we examined aortic valve replacements in vitro. We found that biological valves alter aortic hemodynamics similar to bicuspid valves, whereas mechanical valves allow near-normal hemodynamics. The long-term consequences of this remains unclear. However, more and more younger patients nowadays receive biological valve replacements, and they could actually live to see the consequences of disturbed hemodynamics in the ascending aorta, such as potential aneurysm formation. Since the influence of the surgical procedure itself cannot be evaluated with a silicon model, we developed an ex vivo swine model. Only the aortic access for the valve replacement, without touching the valve, led to vortices at the suture in these aortas. So just the sham surgery already led to disturbed flow in the ascending aorta. That means that not only the valve, but also the surgical access can affect post-operative hemodynamics. In addition to conventional valves, we were able to test new valve reconstruction techniques like the Ozaki neocuspidization in our model. With other doctoral students, we aim to understand the factors that influenced 4D flow MRI measurements, including during the acquisition, the analysis, and physiologic variability. And surprise, all of the above influence the measurements somehow. For my combined work, I received the habilitation at the University of Lübeck, which is the highest university degree in Germany. During my residency, I was also the trainee representative in the German Radiology Society and founded the Young Radiology Forum. This new trainee organization within the society is now the largest interest group in the society, counting more than 4,000 members with active representatives in every study group. After our board certification in radiology, my husband and I moved to Wisconsin Madison for a research fellowship for 2 years. We joined Scott Reeder's lab and were blown away by the welcoming and excellent research environment and the wonderful people. I've got to work with 4D flow MRI researchers who have been my idols for many years: Scott Reeder, Oliver Wieben, Kevin Johnson, and Alejo Roldan Alzate. Together, we are studying portal venous hemodynamics in patients with end-stage liver disease and portal hypertension. Some of these patients develop gastroesophageal varices that can lead to life-threatening bleeding if not treated. Currently, patients should undergo EGD endoscopy to detect the varices. The ones with varices can be treated directly. However, many patients undergo EGD just to see that they don't have varices. Wouldn't it be nice if we could identify the patients who really need the EGD for treatment beforehand? The research group previously did a pilot study which showed that we can identify the patients with varices at risk for bleeding by indirectly or directly measuring the flow in the contributing left gastric vein. There is a flow reversal in patients with varices, whereas there's no significant flow reversal in patients without significant varices. In the past years, we have been refining this method further, and we are currently enrolling a decent number of patients to confirm the pilot findings. If successful, a 3-minute 4D flow MRI could be added to routine HCC screening in these patients instead of an invasive EGD. Here are typical examples of portal venous flow in a healthy volunteer. Here you can see a patient with gastroesophageal varices at high risk for bleeding as determined by the EGD, and there is a flow reversal in the left gastric vein. In the middle, there is a patient also with gastroesophageal varices, but that were determined as not high risk. And there you can see no significant flow reversal in the left gastric vein. During my time as a visiting professor in Madison, I was named Junior Fellow of ISMRM. That gave me the opportunity to organize the wonderful secret sessions with a colleague. Additionally, Ellen Grant, who is wonderful, became my mentor, and I was elected into the board of the Flow in Motion study group. After 2 wonderful years in Madison, I asked Scott Reeder for his advice, which offer in Germany I should accept. His simple question was, "Why don't you stay here?" Scott convinced my husband and me that UW-Madison would be the most wonderful place on earth, and it would be worthwhile repeating the major med school exams 10 years after graduating and the radiology board exams just to stay here. And he was right. In the beginning, it sounded a lot easier than it actually was. Repeating the med school exam 10 years after med school while working in research and having a small child at home with your husband doing the same thing wasn't always fun. Luckily, we didn't know that beforehand, and we are now more than happy that everything worked out. After a 1-year cardiothoracic imaging fellowship, I now started my faculty position and a tenure track earlier this year. I am the co-director of our local 3D post-processing lab, Radius, and I've started building my own research lab again, focusing on cardiovascular 4D flow MRI and building upon my prior research. And with that, I would like to thank the SMRA again for this incredible honor. Thank you to my mentors, and especially Jörg Berghausen, Scott Reeder, Prashant Nagpal, and Oliver Weben for all your support. Thank you, Dr. Schiebler, for recommending me to apply for this junior fellowship. Thank you to all my colleagues and friends who supported me along the way. And of course, thank you for your attention. Please feel free to reach out via email if you would like to get in contact and maybe even discuss some research projects together. I hope you have a wonderful time at SMRA. Bye.
Play the next recording.
Hi, I'm Lexi, a postdoc at Northwestern University. Unfortunately, due to visa issues, I am unable to attend this conference in person. I would like to thank the organizers for giving me the opportunity to share my recent project remotely, which is myocardial blood flow quantification in patients with a CIED during stress and rest using a wideband perfusion pulse sequence, an initial feasibility study. Nowadays, over 3 million Americans have CIEDs. Patients with CIEDs presenting with new or worsening cardiac symptoms typically undergo a combination of vasodilator, cardiac and resting echo as part of their clinical workup, which can be inefficient. Alternatively, CMR offers a comprehensive evaluation through various image types such as perfusion, cine, angiography, mapping, and LGE as listed here. There is growing evidence supporting the safety of CMI in patients with CIEDs, including during vasodilator stress testing. However, a key challenge remains, that is, off-resonance artifacts caused by the CIED generator can significantly limit the diagnostic yield of standard pulse sequences, particularly in the context of quantification of MBF and MPR. As shown in the left column here of the phantom data, the signal intensity is significantly changed due to the artifact produced by the ICD. To address this, wideband pulse techniques have been proposed and demonstrated in LGE, T1 mapping, and perfusion to reduce the artifacts as shown in the right column of the phantom data. While previous studies demonstrated that a wideband perfusion pulse sequence could suppress image artifacts for MBF quantification at rest, its utility during vasodilator stress has not been validated. Thereby, the purpose of the study was to evaluate the feasibility of MBF and MPR quantification using a wideband perfusion sequence during adenosine stress in ICD patients. We prospectively enrolled 21 patients with an ICD and performed an adenosine stress CMR protocol using our proposed wideband perfusion pulse sequence. As a control group, we retrospectively identified 11 cases who underwent a research adenosine stress CMR with an ICD tapped below the left clavicle using the same image protocol as the ICD patients. As summarized in the table, the cases in control group had a negative stress perfusion CMR, negative coronary artery disease, and no cardiac event 1 year post-CMR. In addition, the mean biventricular functional parameters in the control group were within normal limits, and the EF values in both ventricles were significantly lower in the patient group compared to the control group. Our proposed wideband perfusion sequence acquired one arterial input function plane and at least 3 short-axis myocardial perfusion planes, both during stress and at rest. The table here summarizes the related parameters, and due to time limit, I will not go into details here. We use compression sensing to reconstruct both the arterial impulse function and myocardial plane images, which was performed in line using the Siemens file framework. We then quantified pixel by pixel stress and rest NDF maps using the steps shown here: respiratory motion correction, proton density normalization, signal intensity time curve adaptation, Galilean conversion, forming model decomposition to finally get the pixel by pixel stress or rest NDF maps. After that, we manually segmented the maps to get the 16 AHS segments, and the MPR is the ratio of mean stress NDF over mean rest NDF. Here we show the stress and rest perfusion images in 3 short axis planes of a representative simulated ICD case from the control group with the corresponding pixel-wise stress and rest NDF maps. This slide presents the stress and rest perfusion images of an ICD patient with hypertrophic cardiomyopathy. The perfusion images demonstrate diffuse subendocardial reversible perfusion defects, especially in the regions with hypertrophy. The maps demonstrated diffusely reduced stress MDF, which was worse in the territories with visible perfusion defects. Coronary CTA demonstrated no obstructive coronary artery disease, indicating that the perfusion abnormalities were due to microvascular dysfunction in the setting of HCM. The wideband perfusion sequence produced high image quality in both groups. As summarized in the table here, there were no significant differences in individual image quality scores or some feature scores. The global mean stress NDF and MPI values were significantly lower in the patient group, while there was no significant difference in the global mean rest NDF values. Similarly, the mean segmental stress NDF and MPI values were significantly lower in the patient group compared to the control group in all 16 AHS segments, while no significant differences were found in mean segmental rest NDF values, except in the apical inferior wall. To summarize, this study demonstrated that it is feasible to quantify stress rest NDF and MPR using wideband perfusion sequence in ICD patients. The significantly lower stress MDF and MPR values observed in the patient group compared to the control group are consistent with differences in their clinical profiles and biventricular functional parameters between the two groups. The stress rest MDF and MPI estimates from our control group with no coronary artery disease and normal cardiac function align with findings from other CMR studies utilizing Fermi modeling. This study had several limitations, which are warranted to be solved in the future. First, the control group was not comprised of matched healthy volunteers. Second, the overall sample size was relatively small. Additionally, the diagnostic performance of MBF and MPL quantification were not validated against invasive coronary angiography or PET. Another limitation was that post-processing required manual labor lasting about 2 hours per patient. Thank you for your time, and if you have any questions about this project, please feel free to reach out to me by the email here.
Okay, so that concludes our Junior Fellowship Award winners talks. Next, we'll be moving on to our invited speakers. So first up, we have, well, maybe I should introduce my co-moderator first, Dr. Emoke Shirinos from Semmelweis University.
Thank you, everyone.
Yes. So our first talk will be by Dr. Kim Lian Nguyen from the University of California, Los Angeles. And she'll be speaking on leveraging an outsider's perspective.
Great. Thank you so very much. Good morning, everyone. Thank you for making it here early in the morning. First, I'd like to thank the organizers and the ECC committee for this invitation. It's a bit daunting to give this presentation because of the many giants in the field of MRA who are in attendance and who have far greater insight about science, work, and life in general. I consider myself a mid-career physician and scientist, so I still have quite a ways to go. And with that in mind, I will defer the advice-giving portion to Dr. Martin Prince, who has had an illustrious career. I will focus on areas that I found less clear, which in so many ways had to do with embracing an outsider's perspective in a winsome way. So I'll share with you my journey, and then I'll spend the rest of the time to provide a few clarification about a few expectations for the early career clinician researcher. And I'd like to share some tips and tricks that worked for me. So early on, I studied chemistry as an undergrad. Then I went to NIH to figure out whether an MD PhD was the right path for me. I worked in a viral genetic engineering lab and did wet work. Although I excelled in it, it was not quite my calling. So then I decided to spend the next 10 years in clinical training, went to medical school, did clinical training in medicine, followed by cardiology. And it was really not until the end of my cardiology training that I discovered Dr. Paul Finn's cardiovascular MR images. And that was a turning point for me because it really made me think about image formation, and that led me to really desire a career in research. And so I went to the NIH to learn more about MRI. I was there for 2 years and worked with Andrew Arai, Peter Kelman, Bob Balaban, and Dr. David Blumke. I came back to UCLA in 2013, teamed up with Dr. Finn and Peng, and began working on pheromoxyl-enhanced MRI and 4D imaging. There, I learned principles of image reconstruction and some signal processing. So I'm a practicing cardiologist who later became a physician scientist because I was very much inspired by the work of a radiologist and found purpose in imaging science and MR physics. I was also a late bloomer when it came to research training and becoming a scientist. So relative to all my peers who knew exactly what they needed to do and how to pursue a research career, I was starting at the end of my clinical training. But I turned challenges into opportunities, and I'll talk about some of those examples here. So about 2 years into my faculty position, while working on pheromoxetol, there was a black box warning issued for pheromoxetol. At that juncture, it was early enough in my research direction that I could turn around, do something else. But I persisted, and I worked very closely with Dr. Finn and Peng to create the FairSafe registry and consortium. And as I continued to work, I was successful at getting my VA merit grant, which is considered an R01 equivalent in MRI and microvascular disease. And shortly thereafter, but right before COVID, I landed my very first NIH R01 on pheromoxyl-enhanced MRI for patients with kidney disease. At that time, COVID started, and so everything shut down. It was very difficult to recruit postdocs and graduate students, yet I persisted. And through those years of the pandemic, I just hunkered down and got a second R01. And that had to do with pheromoxyl-enhanced MRI in intramyocardial hemorrhage and heart failure. And during that time, I also worked to renew my BA merit. And so we kept going. Fast forward to 2025, UCLA recently lost some of its funding, and its funding has been suspended. Fortunately, our lab was not affected. However, I needed to have creative strategies, so I've decided to diversify our assets and funding sources and focus on what I am able to control as opposed to worry about things that I can't control. So I continue to work on Ferromoxyl enhanced MRI with our FairSafe consortium. Our group recently was able to successfully petition for an MR contrast agent study group through ISMRM. I continue to work with Dr. Finn on our 4D music work with congenital heart disease. At the same time, I'm running an NIH-funded lab with trainees consisting of both PhDs and MDs, and I've built out 2 areas of investigation related to fractional myocardial blood volume mapping with ferumoxytol for multi-scale modeling of small vessels. And more recently, a collaboration with Anthony Christodoulou to represent cardiac motion in arrhythmias. And along the way, I've served as a mentoring faculty on NIH T32 grants in areas of imaging science, bioengineering, and cardiovascular technology development. I currently serve as a primary mentor on a VA Career Development Award for clinical scientists. So I just feel very fortunate that I am a part of a much larger interdisciplinary group of researchers who are very encouraging and supportive. So I've talked a little bit about my journey. I'll spend the rest of the time clarifying a few expectations that I myself found confusing, and then maybe share some tips and tricks that worked for me. What does it mean to begin with the end in mind? For an outsider looking in, often we have fresh perspective on what's happening inside. And for me, my first SMRA meeting was in 2015. At that time, I was introduced to Dr. David Saloner, Dr. Edelman, and Dr. Martin Prince. I was terrified. These were names that I read many articles on, but never really thought that I would have the opportunity to meet them. Anyway, by the end of the conference, I felt very comfortable and made up my mind that this was the scientific community that I wanted to be a part of. Fast forward 2025, I'm here saying hello to Dr. Prince and giving the talk before he is giving. So I feel very, very fortunate. It also means recognizing that the currency in academia is grant funding and publications, and I just have to rise to that occasion, whether I like it or not. And then being honest with myself in terms of how much do I like writing, and can I be disciplined enough to set aside time for writing, because that is very crucial. And then realizing that the skills needed to excel as a PI are very similar to those needed by a CEO. So I think that's what it means to begin with the end in mind. And then I've been told to pursue my passion. It's a little bit confusing, but I think what it really means is to commit to something that you're good at and then relentlessly dedicating time to sharpen that skill or set of skills. Committing to a scientific line of inquiry means focusing and having a range of projects that are easy, medium, and hard so that you're always prepared when opportunities arise. And then periodically assessing your career trajectory. Are my choices still the correct ones? So every 4 years, I reconsider my path, and this typically coincides with grant funding duration. At the end of the day, it is my race and it's your race to run, no one else's. So you have to pace yourself. It's a marathon, it's not a sprint. And then the question of what is impactful research? I'm encouraged to do that, but oftentimes I don't really quite know what that means. And so I found this nice pyramid that helps me to consider my research questions and areas as a whole. And I think this paper was authored by Dr. Narula, and I think he should be speaking as a mentor later on today. So maybe you can ask him about this pyramid in terms of what is most impactful. And it seems to me like the most game-changing ideas are the ones that have patient impact. And so I've learned to conclude that hypotheses and discoveries must stand the test of time and reproducibility, whatever it is that you're working on. The tools that you're creating must have purpose. It is probably not a good idea to create a tool and then go around looking for a purpose. It should be the other way around. And then relationships with industry scientists and commercial entities can really widen your sphere of influence. So I find that that relationship actually has helped me to grow. And then I was told that I should manage up, down, and sideways, but I never really knew why. In hindsight, it is because I am my own CEO, my own COO, CFO, and brand consultant. So to do that, I had to figure out the job for which I was hired to do. That meant asking a series of questions of how I can serve. What does my chief need from me? What do my trainees and peers need from me? What does the department need from me? And then defining and creating that job. And some aspect of that has to do with setting expectations and then learning to engage and collaborate with my peers as opposed to competing with them. And then becoming independent. That was also very confusing for me. I noticed that grand success often relied on your scientific hypothesis, the team, infrastructure. But then also, when you look at impactful publications, they often come from a network of multidisciplinary researchers working on complementary science. So why independence? Oops. So I think being an independent investigator really means having the ability or gaining the ability to bring together a synergistic and cohesive team that can deliver on the scientific hypothesis, and then having sufficient scientific prowess or gaining that prowess to craft impactful and testable hypotheses. And lastly, work and life balance. That did not work for me. I found that it was far easier to integrate work into life. And in order to do that effectively, I had to think about resource allocation. How I spent my time, my energy, and talent ultimately shaped my overall life strategy. For a physician scientist, it was helpful for me to think of myself as a scientist first and a physician second, but at the same time to not let go of my clinical insights and remembering why I do research. I do research because of my patients. And then when negotiating with my chair, I had to remember that I am the only person who can protect my time for research. So in summary, leveraging an outsider's perspective meant I had to have a roadmap. Because of that, I learned to craft a strategy for my career and my life. I worked to get good at writing and pitching my science, which is what I'm still working on. I used the early career period to define my expertise and to figure out the job for which I was hired. I had fun along the way by cultivating intentional relationships and coming to conferences like SMRA. I also learned to integrate work and life. And so I feel very fortunate, and I'm so very thankful to the giants in my life, and particularly Dr. Finn, who is really the turning point for my career as a physician and as a scientist. So thank you.
And now please welcome a pioneer, Dr. Martin Prince from Weill Cornell Medicine and Columbia College of Physicians. Dr. Prince will give us an insight on what it's like to be at the beginning of one's scientific career. Dr. Prince, the floor is yours. Thank you.
Okay, thank you so much. And it's fabulous to hear those really fantastic talks from the junior fellows. And to my surprise, to realize I have nearly the same story as Kim Lien, who gave that just fantastic inspirational talk we just heard. So I also want to share with you a few tidbits about how to have early career success, and maybe success that can last your whole career. And I also want to tell a little bit about my story on my early work developing contrast-enhanced MRA. But I want to make a few points I started out actually doing my PhD in laser physics, and this is me back in 1989, finishing up my dissertation, working in the laser lab with a vascular surgeon. And we had the idea to burn the crud out of arteries, known as laser angioplasty, which became completely obsolete with the invention of the stent, which was just like a far better solution to this technical problem. And I found myself having just finished graduate school with no career and no prospects. And incredibly, I had a bit of grant funding, but I had nothing to spend it on because that field had completely imploded. I started my residency and was just wondering what I was going to do with my life. And lo and behold, Dr. Jim Pochin, founder of the MR Angio Club, which we now call the SMRA, came and gave a grand rounds and showed these amazing pictures of the carotid arteries obtained with MR using 2D time of flight, no injection of contrast needed. And at that moment, I realized, oh my God, I'm going to work on this. This is so revolutionary. And at that moment, I was working full-time as a resident, but I was able to get some research time allocated by the chairman because he realized I was so excited about this concept. Now, in this talk, I want to just discuss a couple of things that I learned doing the PhD that I thought were helpful. But later on, I realized that I hadn't fully learned these lessons. So I just put that you might learn these doing a PhD. And this will save you a lot of time because once you learn these, you're kind of pretty much all set. Keep a lab notebook. Do a literature search at the beginning of your project. Work on something that inspires you. And I think we heard that from all of the talks so far, that passion and inspiration that was focused on the project. Take initiative and personal responsibility. Be honest and have an open mind. Never ever try to fudge your data or tweak or twist. Feel confident that you're going to be lucky and make a great discovery. If you are not having that open mind, you're going to miss out on your life opportunity to do something amazing. And start writing your paper at the beginning of the project. All right, so of course, show you a few pages out of my lab notebook. They teamed me up with a physicist. And at that time, our hospital had just acquired this super fast gradient system that enabled echoplanar MRI on those old ancient scanners. And I was assigned the task of figuring out how to synchronize the time of flight MRA with the ECG. So they handed me a copy of the pulse programming code, which was the size of the telephone book. And I realized, wow, the telephone book isn't printed out anymore, so you guys don't know what I'm talking about. But if you think about the Encyclopedia Britannica, if you ever seen that, or anyway, it was a very thick bundle of paper, maybe 10,000 pages of code. But by some miracle, I was able to integrate an ECG gating scheme, and we were testing it out with the physicist that I was assigned to. I'm not going to tell you who it was, but someone very famous. And as we were testing that out with our oscilloscope, plugging into the gradient outputs, I could tell something odd was going on. And very shortly, I realized that the physicist that I were working with had realized that there was some bug in the pulse sequence code that they didn't fully understand. And they were debating among themselves how they were going to get away without having to retract the previous 6 papers that they had submitted. And at that moment, I realized there's no way I can accomplish anything standing on the shoulders of idiots. So fortunately, I had already been working on vascular disease because I did my PhD on laser angioplasty. And I just felt that the way you take pictures of arteries is to inject contrast and then take the picture. That's like how it's done. And even though that was obvious to me, it wasn't obvious to anyone in the MRI field. Well, back in those days, the use of MRI was being limited so it wouldn't cost a lot to society by restricting one MR scanner per city. So in the city of Boston, we had one MRI scanner. And of course, it was really busy and you couldn't get any time on the scanner. And the hospital was able to purchase the right for another scanner in the city across the river called Charleston. Can you see this pointer here? No, okay. We have a pointer? Oh, this guy? All right. Anyway, there was another scanner in Charleston. Of course, it was a big hike to get over there, but I had a bicycle. So I volunteered to ride my bike over there and supervise the cases so that I could figure out how MR worked and tinker and try to figure out how to do the MRA. Well, after I had done a bunch of cases, I thought I would try the gadolinium, and of course it didn't work. And when I did a literature research, I realized that it had already been figured out that it didn't work. And this is one of the papers that Giles had showed us at the opening session, where Creasy reported that when you inject the gadolinium, it just makes everything enhance, and then you don't even see the vessels quite as well as without gadolinium. And there was another example that Giles showed from Graham Bitter. And also, this is something from Bill Bradley, who was a giant in the field because he had written the only book you could read describing how to do anything with MR. Unfortunately, as gadolinium distributes from the intravascular space to the extracellular space, background signal is increased, decreasing the quality of the MR angiogram. And here's Graham Bitter. MR angiography requires no contrast agent. And I do want to say that when you select your laboratory notebook, it should be a bound notebook and it should have numbered pages. And this makes it a legally acceptable documentation of your work if you ever get into a patent battle over whether or not you were the first to have the idea. All right. Well, at that time, there was a famous company called Kodak, and they foresaw, they were making a lot of X-ray film and doing great business, but they foresaw that the digital revolution was coming. So they started trying to use their compounds to do other things. And one of the things they wanted to see if they could see their compounds on MR. So they asked the hospital, oh, can you test out some of our molecules? And I volunteered to help test them out. They assigned me a slot on the Charlestown research scanner. It was very busy, so I was a junior person. They gave me the slot from 2 a.m. to 4 a.m. And they also gave me a technologist. And when I showed up, and they gave me a bunch of rabbits. So when I showed up the first day, we put the rabbit in the scanner, and I said to the tech, oh, okay, let's scan. Well, it turned out it was an animal tech. It wasn't an MRI tech, which was great because the animal tech knew how to start an IV in a rabbit, which I couldn't do. But we just pushed all those buttons and eventually figured out how to get the thing to work. We took some beautiful pictures. And I eventually figured out we needed to image during the injection of the gadolinium rather than inject and then image. But I did some modeling and I figured out that in order to make that work, to have a high enough injection rate with those really slow scanners taking roughly 5 minutes to do a scan, you needed a triple dose, which, back then, a triple dose, first of all, that was a lot of money. And how could I justify that? Well, incredibly, almost at the moment I figured that out, Prohance came out FDA approved at a triple dose. And boom, I was doing it on those cases, and this is my first case. You could see all that atherosclerotic aorta. And the way I did it, of course, we didn't have large syringe because we were used to doing single dose. So I had to load up, usually 2 bottles of gadolinium was enough for a triple dose. So I would load up 2 bottles in 2 syringes, and then I would load up saline flush in the other syringe because I didn't want to waste all the gadolinium that was in the tubing. It went from outside the scanner to inside the scanner. So I would start off with that first syringe, and I'd be pushing that in. And then when I got to I tried to time it so that it would last for the whole scan. Getting the flush done just as the scan was finishing. So I'd finished that first syringe, and then I would quickly grab the second syringe. Oh, shoot. Which one is it? And very quickly I realized it didn't matter which syringe I used for the second one. If I, did the first syringe and then just flush, it came out the same as if it was 2 syringes. And from that, I was able to figure out that the key to this was synchronizing the maximum gadolinium concentration in the blood to coordinate with the acquisition of the center of K-space. And that turned out to be the magic that made the whole thing work and led to lots of further refinements. This is the first MR Angio Club I went to, and these are just some of the people, and there are others who I've seen here who are in this picture. And wow, these people were all giants in the field, inventing some of the core sequences that we're using today. This was my first paper, and very quickly, I had a second paper, the first paper on my first 12 or so patients, and then I got to 100, I published another paper. And at that time, the workstation for doing MIPS and reformations hadn't been invented, so we had to write the code to do that ourselves. Now it's so easy. This is a letter that went to one of my attendings. A quick note of thanks for your timely performance of the MR angiography. Your findings by MR angiography were confirmed at surgery and successfully treated. I was very impressed with the detail of the study, and this was our head of vascular surgery. So it was such enthusiastically embraced by the referring physicians that I became just, fantastic at riding that bicycle over to Charlestown. And we continued to work on developing more refinements. And I'm just going to end with this slide here, a quote from Benjamin Franklin, which I particularly like. Either write something worth reading or do something worth writing about. And I wish everyone great success with your careers. And I know many of you have already been incredibly successful. Thank you very much.
Unfortunately, we're out of time, so we have to move on. But Dr. Wynn and Dr. Prince will be at the coffee break in the seminar room. So please, everyone, ask your questions there. Thank you.
All right, we're going to move on to the next part of our session, the fireside chat. I'd like to invite our fireside chat speakers to come join us up here and our next set of moderators.
Morning, everyone. I would like to invite our three panelists for this fireside chat. So we have Jagat Narula from the University of Texas. He's a MD, PhD, cardiologist, and president of the World Heart Federation. We have Hideki Ota, professor at the University of Mie in Japan, MD, PhD, and radiologist. And we have Trisha Roy from Houston Center in the United States. She's MD, PhD, and vascular surgeon. Thank you very much for being here. So during this session, we will try to get to know better our panelists. Also, this is the opportunity to share with all of us anecdotes and anything you would like to share. And also the audience is very welcome to ask questions to our panelists. So I think we will start and then I hope you will jump on in the discussion.
Okay, I have a question. What strategies have you found most effective in mentoring imaging scientists across cultures? Okay. What strategies have you found most effective in mentoring imaging scientists across cultures? Like different cultures, how can you manage scientists?
I think a lot has been said already. First, Dr. Nguyen, she presented it, and then Martin talked about it. And I think first important thing is that you have to relentlessly seek a mentor. So I do not know whether it is the mentee which finds the mentor or the mentor that finds the mentee, or why is it or how is it that the combination occurs. But it is extremely important. And as you see in Dr. Nguyen's case, she has worked with people like Paul Finn and David Blumke and Andrew Arai and Balaban and all those. And similarly, he started with Dr. Pochin and all. I mean, I think, and I do not know whether Martin remembers, we overlapped each other at Mass General for almost 7 or 8 years when we were there together. But you know, it is extremely important. And then one important thing that he said: prepare yourself well. So the strategies, I don't think there are much of strategies here. You have to work towards it. You have to try to find the right kind of a mentor, something you like, read about it, go to the conferences, prepare yourself, and get ready for something what you really like. I don't think there are too many strategies about it. And again, as Dr. Nguyen said, it has to be something that you are passionate about.
Yes. From my career, that I had a chance to go to the I now work as a clinical radiologist in Japan, and doing mainly the clinical things and also some research about MR and CT and interventional radiology as well. So, with this my career, I just had a chance to go to the US to see the University of Washington, and there was the Professor Chong Yang and doing the carotid research. And because from my career, I'm interested in vascular diseases and cardiac diseases, so that luckily, I was just lucky at that time that they are recruiting postdocs, so that I had a chance to go there. Okay, so like you mentioned, I do not have very strong strategy, but I'm really thankful, and I feel it very important to be in good connection with the people and the researchers I met at the time, and I just keep on, communicating and making some the research topics relating with those kind of great people, and then I will try to find out them with my interest, and I try to find out the new topics and interesting things related to the where I can do during the situation in my career. Okay.
I just wanted to add a quick note about the across cultures piece of it. I think that that's a really interesting question, actually, because it is a bit challenging. So I'm a 50-50 vascular surgeon scientist, and so I'm pretty new at mentoring trainees. But like Judith is one of my mentees, and she's Hungarian, obviously from here. Same with Inigo. And it took a little bit of time for me to realize that, she's very quiet and a little bit understated but brilliant, right? And I'm used to, like, now in Houston in Texas, you have to talk with confidence and, it's a very different kind of culture. So I think that that's been really exciting, actually, because I think that there's like a different approach that they're taking to how they're analyzing images and gave me a new perspective. And hopefully, can help them with their confidence and communication style to be more, reflective of the U.S. audience. I think that the cultural aspect of this is actually an important question and something that I'm still very much learning. I know that you've mentored a lot of Japanese mentees, for example. So I'd be curious about if you had any thoughts on that piece of it too.
I tell you that I've had the opportunity of working with Trisha for almost a year now, and I think she's an exemplary model for the young mentors and the way the people flock around her. But one thing I would really like to add from the Japanese perspective, there are approximately 40 Japanese fellows that have visited my lab and have done their PhDs. And almost 30 of them have won the Young Investigator Award at various national and international conferences and competitions. One important thing that I've really seen, that dedication, that passion, and specifically when I see Hideki also, I realize that he, having worked with the carotids and all with Chan and Hatsukami and others, the important part here is that this dedication, they all starting to get together and all. There is one thing which I realized is that when you are a mentor, and if you are working with them, and I have seen that in Trisha a lot, how much are you available to them? How much time can you spend with them? They are there. So what used to happen was that, being clinically involved all the time, significantly, was not able to spend that much of time in the lab, and these people are working. So what we did was that on Saturday mornings, they used to come, these Japanese fellows, at 4 o'clock in the morning at home. So I wake up at 3 o'clock every day. And so 4 o'clock was the time for them to come. And these dedicated, passionate guys, they will come at 3:45. They keep standing outside and 4:00, they will press the bell at home. And then they will come in and we will have our breakfast together. We are writing with them. We are preparing slides with them. We are writing their papers. We are writing their grants and all. And they will leave at about 1 o'clock at the lunchtime. That was every single Saturday, the sacrosanct time. Nothing else will happen. And my thanks to my wife, who just gave up on me. And but I tell you that it is all about the passion. It is all about getting together. It is all about working hard. But you being able to spend time with them. If you accept somebody as the mentor, who's as a mentee, somebody who's spending his 2 years of his time with me, not taking a dime from me, being supported by his country or from his institutions and all, I have absolutely no business not to be available to them.
Thank you very much for the great answers. Any questions from the audience? Okay, I have another question. Each one of you has taken a unique path to arrive at the forefront of cardiovascular medicine and imaging. Could you share one defining moment or decision that shaped your career trajectory and still influences how you think about your work today?
Yes. Because I thought that I will have to just start looking at multiple such defining moments of my life. And I was just trying to figure out as to which one should I choose to present if I really have to talk about one. I think if I have to pick up one, I was, I would say that the paper or the study that really made my life, and that was overnight after the paper was published. That was back in Mass General, 1996. And it was on apoptosis in heart failure. And something at that time, it was becoming very popular, very trendy, because although the apoptosis was described back in 1970s in breast cancer and other areas, but they always thought that the organs that are not able to reproduce their cells, quite like heart and brain, they should not be capable of committing suicide. And it was kind of a taboo that you cannot have an apoptosis, and the apoptosis does not occur in heart or brain. And somehow or the other, my wife, as you will be hearing her in the afternoon, who is a pathologist, sitting with her when I used to see the endomyocardial biopsies back in Mass General, when I was doing my transplant heart failure training. And at that time, I would look at those dropout cells in the cardiomyopathy, and I will always get excited about them. There is no inflammation around there, but you can see the dropout cells there. Looks like that these are apoptotic. Possibly must have been an apoptosis. However, when I talk to people around me and they say, oh, don't even go there. So question was whether you just stop there, follow the dogmas. Who makes the dogmas? We. So should we stop there, or should I really go ahead? Talk to my mentor, who said, you know what, you always think about the crazy idea. And I sometimes support them, but sometimes I feel that I'm wasting money. But somehow or the other, looking at my enthusiasm allowed me to do that. We started working on it, and here it is. We come up with the New England Journal of Medicine paper in that, and that was start of a career, because Boston Globe, a full page coverage, New York Times, full page coverage, whole day CNN is showing heart muscle cells commit suicide. The fact was not there. The fact was that the people who were entrenched in that area were a little, I don't know, not very happy or whatever else it was. There was a lot of criticism that, oh, you know, he's showing so many large number of cells in the endomyocardial biopsies. And if it is so, the person should have died within 6 days' time. Could have been possible, because these were all explanted hearts on which I did, and these people were working on animals. So there was a lot of difference in what I'm looking at and what they are looking at, but they said that my numbers were not right. Went to Dana-Farber for looking at the upstream cascade, because I wanted to prove myself right. I did not want to fight with them. They were big investigators who were criticizing me, so you just cannot go against them. So now here is the story, which is extremely, extremely important, which you must look at it from a very different perspective. So I go there, I start working on the upstream cascade with one very famous person in Dana-Farber. And I look at it, absolutely every single thing is proven, that you have got the caspase 3 activated, you've got all the proteolysis going on, there is a vacuolization of the cells which is occurring, myocytolysis which is occurring. However, when I look at the nucleus is not fragmenting, which is the end of apoptosis. And I remember fighting with my wife also, saying that you can't find me a single cell which will make our paper go to Nature or Science. And she has like thousands of electron micrographs. I talked to Dr. Eloisa Arbustini from Italy, who's a superb pathologist. I get her help. She's not able to find a single nucleus which is fragmenting. So I go to the mentor, and in Dana-Farber, whom I had adapted. I used to do my clinical work in the day. 6 o'clock, I'll go to his lab, work through the night, come back in the morning, do my clinical fellowship again, and night I'm back in the lab. Everything has happened, and I told him that the nucleus is not dying. And he says, okay, you came here to learn upstream cascade, stop at upstream cascade, don't talk about the nucleus. Why should I not talk about the nucleus? Oh, because it does not fit in. He called multiple investigators, and they said he's wrong. If cytochrome C is released, if caspase 3 is activated, The cell has to die, nuclear has to die. Otherwise, it is the wrong. So he said, just don't present it. I'm not going to do that because it is as bad as falsifying the data. He threw me out of his lab. I came back to Mass General, talked to my boss, and Martin will remember him very well. He said, I will support you, go ahead and publish it. I said, I don't care whether it goes to Nature or it goes to Journal of Cardiac Failure. I'll just publish the right findings. The paper went to PNAS. It became a landmark. John Reed, who is the father of apoptosis in United States, wrote that this is a paper of paramount national health importance. And it should become a gold standard that anytime you want to describe the apoptosis in heart or brain, you must show this nuclear fragmentation may not occur. And if the proteins are gone in heart, but nuclear blueprint is intact, everything can be reconstituted and cell will come back. That was the start of reversibility of heart failure. And then that led to our demonstrating that we can identify apoptosis by imaging in heart in a living human being. And that was our next Nature paper, which carried a beautiful editorial by Richard Kitsis, which said, seeing death in the living. So if you want to call one as dogmas, don't believe in dogmas. Be true to yourself. Nature is much smarter than you are. You may think, don't try to fit in your hypothesis or the results into your hypothesis. See what is happening. Learn from there. Try to explain that. And if you fail to do that, keep pursuing it. But be honest with yourself. So that is my divine.
Okay. In my case, my story would be not as big as you did. Okay. But okay, so in my early career, when I was a resident, I worked. I was in a community small hospital over there. At the time, the multi-detector CT is just on the market at that time, and my boss over there, Professor Kei Takase, who is a Japanese guy. And he was the first person to visualize the artery of Adam Kubich, which is originated from the segmental arteries. And so using the multi-detector CT. So it's verifying the vessel, as you know, the very small vessels supplying the spine. And I'm really shocked. So at the time, even the acute appendicitis, it cannot be seen on CT because of because of the limited spatial resolution. But the MDCT makes it possible to see very fine the structures. And so I'm really happy to see we can see the very small things, very vascular, small vascular structures. So, and also I'm an interventional radiologist doing the treating patients with the vascular diseases. So the thing is that I'm really interested in, I became interested in looking at what's going on about the anatomy of the vasculature, and also seeing the treatment effect after our treatment or the treatment by my colleagues and so on. So basically my boss is a nice mentor for me to having the interest in, and I'm always curious about how we can tell the treatment, how we can tell the conditions of patients with various types of vascular diseases. So these are kind of my motivations. And
Can I add one thing to yours? I'm also very lucky here because we used to look at the plaques which are vulnerable to rupture and result in acute MI and sudden deaths and all. And again, looking at all the pathology, again, I'm very lucky having seen all the pathological specimens with my wife and also working with Dr. Renu Virmani at that time and looking at the high-risk plaques. I had an idea that I would be able to show it by CT. And every single person laughed at me when I talked about it. And hey, come on, do you think that you'll be able to look at the vulnerable plaques? I'm looking at pathology, sure, but there's no point looking at pathology. I really want to see it. And this is what the example would be in the afternoon when Trisha presents her MR images on the peripheral artery disease. So I couldn't find somebody who would believe in me. I'm not a radiologist. I had an idea I would be able to do that. And we only had 16 slice CT at that time, and started to look at it, started to find something that I can see the low attenuation plaques, can look at the positive remodeling probably, which is what I used to see in pathological specimen. Nobody would believe in me at that point in time. Ended up in Japan. Went to Dr. Takeshi Kondo in Nagoya, in Fujita University, and stayed at his home for 2 weeks, learned with him, and then, thanks to Toshiba, that they gave their first 64 slice CT when it came to United States. It was given to me as a gift. And we started working on it. And after that, it's all history. 3,000 patients, 10 years follow-up. We literally wrote down the rules of plaque vulnerability. But yes, it all started with the advent of the advanced imaging tools that really make it happen. And now you have got much better technology at your hands with 600 slicers and 0.25 millimeters slice thickness and whatnot. But absolutely.
I'll be a bit quick, but so I did my PhD. So in our program in the University of Toronto, we did 2 years of vascular surgery, and then I stepped out for my PhD and I did it in a Graham Wright's lab. And I just remember the first time that Gary showed me one of the postdocs in his labs, showed me how to use a 7 Tesla and image these ex vivo vessels. And he showed me the first set of pictures of this ultra short echo time imaging. And I'm like, what is that? And it was a popliteal artery with all this speckled calcium and this dense collagen. And I'm like, what is that? Like, I've never seen anything like that. And it made me just realize, like, wow, we are completely blind with what we're doing. We're developing all of our imaging, all of our treatments and all of our devices. And we have no idea what these plaques actually look like and what they're made out of. And the fact that you could use MRI to, I'm a materials engineer by background, and the fact that those MRI characteristics can actually relate to material properties, man, it was mind blowing. But seeing that first picture, I was just, I was hooked. That was awesome.
Thank you very much. If you have a questions, please ask the audience. But I was wondering now about the future, because we have the younger generation here. And so how do you see the future of MRA and MRI? Like, what challenges, issues should we work on, contribute?
For MRI, of course, I think that's looking at the vessel itself. Probably because of the yesterday we had the session looking at the photon counting CT and so on, which gives us a very high spatial resolution. But so that I think the challenge would be to see the very fine structures, how we can compete with CT. Well, it's kind of difficult thing, maybe currently, but in the future we don't know. Okay, but because we can see we do have so many advantages over CT, like to see the directly the flow and also having very much better tissue contrast to characterize the plaque components as well. So that I think that we should stick on. I think we can, utilize the advantages of MRI among various modalities for further research, I think.
I'd say the opportunities, and I may be a little bit biased, is in the legs. Okay, so peripheral artery disease. I think that is amazing application for MRI. I don't think CT can compete right now. Maybe photon counting CT in the future, but not requiring contrast and being able to image very small vessels, the vessel wall characteristics. It's just completely unexplored, and I think that there's so much potential in the arteries and the veins and the lymphatics. Like, I think that this is something that is just ready to explode. And so for future scientists, I would very much encourage you to look at the legs.
I think I'm not too much of an MR person. So I will be, I'll keep my comments limited, but I do believe there are certain areas, specifically when we talk about the cardiovascular diseases, which I really feel are extremely important. Cardiomyopathies, first of all. I think it's a must that we should have one MRI in a cardiomyopathy in the person's life, at least once. The Mojs classification, which we propose from World Heart Federation, the second iteration of it, which is coming, which we are actually meeting at European Society of Cardiology for that, with Dr. Perry Elliott and Luis Arbustini and all others, and Limangelli and other folks, that I think would be a recommendation that we must do that as one of the important things. We can also look at the genetic possibilities, that genomic analysis, just by looking at the MRIs and AI. So I think it will be extremely important to look at those signatures. Importance of your T1 weighted images much beyond your gadolinium, the late gadolinium enhancement and all, as far as the outcomes are concerned and other things. So I think in cardiomyopathies, in my opinion, even more than anything else, I think MRI has a big future. But before you ask one question, I just want to compliment you, and I want to compliment the SMRA for what you people do here, bringing for the emerging leaders. I think this is the best thing that I have seen. Does not happen in other societies, and I think they must follow this. When I was the editor-in-chief of JACC Imaging, I had created something what you call as the emerging editorial consultants. Quite a lot of them have now become the editors themselves, not only the associate editors of the journals, and most of them graduated to the full editorial consultancies. But important is that this is what I have seen here. I tell you, this is one of the, it should be complimented. And I'm really honored that I'm sitting here with you. This is something we have to do.
This is really our honor to have such three amazing person around us. And I think we can conclude this session and continue this passionate discussion during the coffee break, where we will have different mentors. And this is really the opportunity for you to meet mentors.